theme antibody drug conjugation in cancer therapy. I think all of this very in line with the current state of health technology. I would like and International Islamic University Malaysia IIUM, speaker Profesor Dr. Muhammad Tahir Bahtiar, Senior Lecturer of Department of Pharmaceutical Technology IIUM in Kuala Lumpur, Malaysia. Let me introduce myself. My name Puji Isyanto, Vice Rector of Universitas Buana Perjuangan Karawang, or UBP, yes. UBP Karawang, West Java in Indonesia. I will give brief profile about the Universitas Buana Perjuangan Karawang. And now, higher education cooperation is very, very important. As I said, it's very, very important towards improving the quality and institutions as graduates. Higher education or universities can apply independent learning on independent campus about domestic or global. In Indonesia, independent learning and independent campus, which is MBKM, Merdeka Belajar Kampus Merdeka, yang kita kenal dengan program Menristek Dikti tentang MBKM, sehingga program kolaborasi collaboration as will very important to universities. Universitas Buana Perjuangan Karawang was established to accommodate the need of the Karawang community for science and technology which are changing rapidly. This is related to local government program in increasing the human development index for the Karawang community in West Java, Indonesia, and play an active role in national building. Universitas Buana Perjuangan Karawang is the capital of the Karawang Regency of West Java, Indonesia. It is very near from Jakarta, about 32 miles east of Jakarta. Karawang Regency is known as a major rice production, sebagai lumbung padi di Indonesia, rice production in Indonesia, in West Java, Karawang. Karawang is also known for its industrial area for manufacturing such as automotive, automobile, electronics, manufacturing facility, health manufacturing, industrial hospital. In Karawang, about 29 hospital in Karawang, including rumah sakit umum and rumah sakit swasta. In manufacturing facilities in Karawang, West Java, Indonesia, including Toyota, Honda, Yamaha, Mitsubishi, and manufacturing the other. As the collaboration conducted UBP Karawang with institution, with company, with universities, this activity provides a benefit for both parties based on mutual benefit and trust purpose. Kami menginginkan kolaborasi ini lebih dekat lagi, tidak hanya sebatas kuliah tamu, ataupun mungkin nanti bisa saling pertukaran, joint research, joint publication. Further collaboration is contained in the draft cooperation agreed by both parties. Some benefits as acceptable as follow: improve. University improve the image of institution or company that actively participate and care about the environment, especially the world of education. And collaboration acceptable participate in the development of a higher education curriculum by the needs of the community and the industrial world. As like this moment in Zoom meeting, guest lecture about industrial drug in cancer therapy, yeah, antibody drug conjugation in cancer therapy. It's very, very important in technology of health technology. Terakhir, kami mengucapkan terima kasih. Thank you for speaker, Profesor Dr. Muhammad Takhir Bakhtiar from IIUM Malaysia, Universitas Bakti Tunas Husada, Pak Dekan Farmasi, Universitas Garut, dan juga Universitas Buana Perjuangan Karawang. Semoga collaboration yang kita bangun menjadi peningkatan untuk institusi dan kualitas lulusan di semua perguruan tinggi yang terjamin untuk mendapatkan kualitas yang diakui oleh masyarakat. University of Buana Perjuangan Karawang supported this agenda, supported the collaboration of facility or support dukungan, ya, support dukungan student internship, development and technology, educational scholarship, establish joint committee and so on. Sekali lagi terima kasih, dan demikian yang dapat kami sampaikan. Bismillahirrahmanirrahim. Alhamdulillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Terima kasih, Pak.
Thank you for your speak, Dr. Haji Rianto as Vice Rector from UBP. And then we can say hello from Uniga. Ibu Dr. Mrs. Dr. Rizka, are you here?
Yes, I am, Pak.
Yes, please.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for the opportunity. Thank you for the attending the guest lecture collaboration, and a special thanks to Prof. M. Taher Bakhtiar. Terima kasih atas kehadirannya.
Sama-sama.
I would like to say thank you too for the UBTH for organizing this event. We hope to get a lot of benefit from this event. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you from Uniga, Mrs. Rizka Prasetyawati. And then next agenda is the lecture we have been waiting for. We proudly present the lecture from International Islamic University Malaysia, Prof. Taher. Assalamualaikum, Prof. Taher.
Waalaikumsalam.
Okay, terima kasih. For the next, I will read about his CV or curriculum vitae. Full name, Prof. Dr. M. Taher bin Bakhtiar, PhD. Prof. Taher obtained his Bachelor of Pharmacy from University of Andalas, Indonesia in 1997, and Master of Science in Natural Product Chemistry from University of Technology Malaysia, UTM in 2000. His PhD in bioprocess was obtained in 2005 from UTM, and soon after that he joined International Islamic University Malaysia. His research interest is in drug discovery involving extraction, isolation, characterization, and bioactivity study of natural products from plants. His current research is wound healing, anti-cancer, anti-obesity, anti-diabetes study of several medicinal plants using animal cell culture. He has published several papers in reputable journal and has contributed chapter in books. Okay, we can for the next few hours. Over the next few hours, this meeting will provide you plenty of opportunity to discuss, share knowledge and insight about antibody drug conjugation in cancer therapy. To Profesor Dr. M. Taher bin Bakhtiar, please.
Ya, baik. Terima kasih Pak Diki. for kind introduction. Juga terima kasih kepada 3 institusi yang sudah bergabung bersama untuk mendengarkan kuliah tamu dari saya yang mungkin little knowledge ya dari saya. Jadi izinkan saya untuk slide. Boleh dilihat?
Yep.
Oke, baik. Oke, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. It is my great pleasure to be here as a guest lecturer at 3 institutions in Indonesia. And I would like to thank the organizer, host for this lecture. So my topic is on antibody drug conjugates, or ADC for cancer therapy. Actually, this field of therapy is quite new. It was introduced in 2011, but before that, there are some research had been done with regard to the use of antibody actually to carry the drug to the target site. This kind of therapy is quite different compared to other type chemotherapy. The conventional chemotherapy is just use drug, chemicals, drugs like tamoxifen, what else, doxorubicin, they need to be taken orally or through intraperitoneal routes. But we know that conventional therapy is quite or what called harmful to the patient. It can actually affect normal cell as well, because no specific action of the drug, because the effect is systemic. It is throughout the body. It will affect other cells and organs as well. So by using this kind of approach, we can actually use monoclonal antibodies to carry the anti-cancer drug that can be released to the site of the cancer. So the basic knowledge of this actually with regard to the interaction of antigen and antibody. We know that antibody is specific, very specific to bind to the antigen. So this kind of approach is quite what called useful. So the problem in ADC actually is quite diverse as well, because not all anti-cancer drug can be conjugate, can be bind with antibody in order to carry them to the target site. So there's some actually constraint and other parameters that need to be observed. So I will try to explain, from the beginning, like A to Z in just a few hours, 1 or maybe 1 hour. I hope that all of you, the respected lecturers and also students, can get benefit from this. But actually, this kind of therapy is quite helpful because it can reduce the side effect of drug to the body. Just only what we want actually just to kill the cancer cell, not to kill the healthy cells and other the tissue. So but this kind of therapy is quite expensive. Maybe not available. I'm not sure whether it is available only in Indonesia. Actually, so far only 2 successful ADC drug that is available, especially for breast cancer and also non-Hodgkin cell lymphoma. This picture is actually example. Actually, the successful one, Acetris and also Kadcyla. Mylotrax not successful. So with regard to the history regarding the use of antibody in therapy, basically antibody itself can be used to treat. But antibody itself is working actually to kill any foreign substance in our body. we know antibody, right? Healthy people will produce it, yeah, in response to any kind of foreign substance. Yeah, foreign substance here can be virus, can be bacteria, can be cancer. Yeah, so actually they are doing their job without our sometimes without our concern. Yeah, because human body is quite Allah actually creates in very perfect ways, but maybe in certain condition we are not able to manage it sample with certain conditions, our body not able to produce in a good amount of any kind of biochemical in the body, including insulin sample. In normal people, they don't have problem with how to metabolize carbohydrate, but certain people they have problem with how to metabolize the carbohydrate. So that kind of actually, so this condition need to be helped. Yeah, I mean, we need to supply from outside. So this actually theory is basically introduced by Paul Ehrlich, in 1909. Actually, related to magic bullets. Yeah, magic bullet means they are going to the target. Yeah, target directly. Yeah, like we are aiming something using nuclear bomb or something. We want to bomb somewhere actually far away from, but actually not going to other place. So this story. And then 1976, Kohler and Milstein already employed actually how to produce a large amount of monoclonal antibody. So basically, what happened in our body? The cancer cell actually is considered as foreign substance. Yeah? Actually, in the cancer that produced by patient, actually the surrounding, the killing cells, the surrounding cell, they produce proteins. Yeah, with proteins we call it antigen. Yeah, sometimes this terminology can be interchanged. Yeah, we know that protein. Yeah, but sometimes can be antigen. Yeah. But the antigen must be protein. Yeah. So what is protein? Protein is actually made by peptide. Peptide made by amino acid. Yeah. So this peptide or protein is specific to bind to antibody. Yeah. So basically, the patient with cancer, example, and other kind of disease, they will produce antigen in their body. So meaning the antigen is just only surrounding the cancer cells. Yeah. And then if we use antibody to detect them, yeah, antibody will be easily to detect. Yeah, I mean they can detect and bind surrounding the cancer cell. And at the site, if we able to bind or conjugate the antibody, that our antibody with drug, anti-cancer drug, we will also allow the drug to be brought into the target and will be released. Yeah. So release there is depend on the condition or environmental condition surrounding the cell. Can be based on peptides. It can be based on what you call acidity. It can be based on the pH as well. Yeah. So we'll see it later. Yeah. So this is actually the basic theory with regard to the use of monoclonal antibodies in drug delivery. So before that, we have to know that actually antibody itself can kill the cancer cells. So, but sometimes they are not able to, not sufficient enough to cancer. Yeah. So meaning in ADC, antibody drug conjugation using monoclonal antibodies, actually what we expect is actually synergistic, synergistic effect of monoclonal antibody itself and drugs. Yeah, drug here is chemical. So let's check. Yeah, we get to the what called the market of monoclonal antibodies. You see the top sales pharma drug in 2020. Yeah, the first one is Humira. You see this MAB. Yeah, MAB refer to monoclonal antibodies. Yeah, so meaning the top sales. Yeah. Top selling drugs in 2020, 2021 not available yet. I tried to find, but this one just quite old, 2020. But what we can see here, monoclonal antibody is the top, yeah, not paracetamol, not what else, what you expect to be the top here. And how to recognize actually, yeah? Humira is actually, this one is for rheumatoid arthritis, yeah. You see MAB, yeah? And at the end our suffix of the name, actually this one is monoclonal antibodies. What else? Keytruda also monoclonal antibodies, yeah. Stelera. Can you see my arrow? I'm showing something using just arrow, not. Can you see? I move my arrow.
Yes, sir.
Yes, yeah. I think I don't need to use pen. Yeah, or maybe you can use pen just to. Yeah. Okay. So you see what else? MAB, Dupixent also. Yeah. So Avastin. So this actually just what call it? Showing us that actually monoclonal antibody is very important in therapy. So why people prefer to use monoclonal antibody in therapy? Because of side effect. Yeah, actually whatever drug that we consume, yeah, chemical drugs, yeah, any chemical drug that we consume, actually they are toxic to our body. Yeah, because they are not actually something that commonly recognized, yeah, by our body. It will affect kidney for disposal, and also liver to metabolize it. Yeah, and then if you use biologic product, yeah, monoclonal antibody is considered as biologic. Biologic means something that is bio. Bio is actually mimicking what actually our body produce. Yeah. So it has less side effect, yeah, to our body. So nowadays people believe that to use biologic drug is more, safe, yeah, compared to chemical drugs, yeah. And then that's why trending now the people to would like to get biologic, yeah, biologic biopharmaceutical product, yeah, not natural product, yeah. Natural product is actually producing chemical, yeah. They are not safe. Yeah, some people said that natural product is safe. I said no. Natural product is producing chemicals. Yeah, even though they are from natural. Yeah, natural mean they are naturals against synthetic. Yeah, this is actually the different way. But the natural product also produce chemical. So no, I'm not against. Let's just compare. Yeah? Not against natural product. Whatever. As we know, natural product is actually the source of the drug nowadays. Yeah. But they are different. Different. And then we get to the market. Yeah? Market antibody. You see, based on the figure just now, it is it was 50 billion. Yeah, so it is quite huge. Meaning, if we, all of us are becoming pharmacy or becoming a pharmacy, yeah? This one is quite important. Field to be default in future, because we gain just very huge of income to the country, also to the company, also they can help people to work in that company. And then related to the use of monoclonal antibodies, yeah, as we mentioned just now, actually there are some benefit of monoclonal antibodies. The first one is to be used in diagnostic. Yeah, actually, if the antibody that is produced by a body can be used as diagnostic. For example, for someone with something infection, whatever cancer, they need to check the amount of antibody titer, yeah, in their body, actually to check what kind of disease, yeah, happening to that people sample. And then monoclonal antibody can be used in research reagent. Yeah, if you are familiar with ELISA, ELISA is actually using monoclonal antibodies. Yeah, so the basic principle of ELISA is antigen antibody recognition. Yeah, so only specific antigen will be recognized by specific antibody. Yeah, so to quantification and quality assessment can be used. Yeah. And also for human therapy, yeah, human therapy means we can use monoclonal antibody directly as medicine. Yeah. And last one is for drug delivery. Actually, this is actually what our topic today, to use monoclonal antibodies as drug delivery system. So the basic, actually, what called the reason why monoclonal can be used in drug delivery. You see this illustration of cancer cells. Yeah, these cancer cells. So the cancer cell is something that I mean weird, yeah, or something that asing bagi badan kita, ya, bagi tubuh kita. So surrounding the cancer cell, they will produce protein, yeah. They will produce different kind of protein specifically, yeah. So specific protein, yeah, is produced by specific cancer. Like breast cancer, they produce HER2 protein, yeah, markernya HER2, yeah. And also for non-Hodgkin lymphoma, they produce CD30, yeah, CD30. So specific cancer cell, they will produce specific type of protein, yeah. That this protein is considered as antigen, yeah, is antigen. And then we can just find the suitable monoclonal antibody to recognize it, yeah, recognize it. And then they will bind, yeah. They will bind, I mean, the antibody, monoclonal antibody and antigen will be binding, yeah. So meaning in this monoclonal antibody, we can attach, yeah? We can add or conjugate drug, yeah. So meaning when ADC has been prepared, or we produce ADC, antibody drug conjugation, meaning the antibody is carrying drug, so we can inject to the patient, yeah, through parenteral administration. So the antibody carrying drug will go directly to these cells, yeah, to cancer cell and bind, yeah, bind. And this because surrounding the cancer cell, they have different condition with regard to the pH, yeah, can be because of enzyme surrounding here. We'll check it later how. So the drug later, because of different condition, the drug will be released, yeah. The drug will be released, but the drug will not be released during the transport, during the travel, yeah. When we inject through blood vessel, okay, so the drug will be go to the target, yeah. So we don't want the drug to be released during the travel process, yeah, before they are reaching the cancer site, yeah. So this actually we have to play with the what called the linker, yeah, linker that can link. The linker must be specific, you know, to react with different condition later on, yeah. So the drug should be released, yeah, during the travel, yeah, when we inject sample through parenteral immunization and goes to the target site, yeah. So the drug should not be released, yeah, during the travel or transport. Let's search about monoclonal antibody itself, yeah. Antibodies, sometimes we call it immunoglobulin, yeah, IG, yeah. Normally we call it IG, yeah. Group of glycoprotein produced by lymphocyte, yeah, beta lymphocyte in response to antigen. Actually, we can make antibody, yeah. We can make antibody. Normally, the therapeutic antibody is produced using mouse, yeah, using mouse, not human, yeah, because we need to sacrifice the the cell, so we cannot use ethically we cannot use human to produce antibody. So meaning the monoclonal antibodies that are available on the market actually originally from mouse, yeah. We create from mouse production, and later on we can modify the structure to be accepted, yeah. Maybe some of you concerned about rodent product. Rodent means product from mouse, mice, what called rats, yeah. What else? Any rodent like hamster cannot be accepted by human, yeah. But we need to do something on the structure of antibody in order, you know, to reduce the immunogenic reaction, yeah, by our body. And then antibodies, with regard to their shape, it is like a Y, yeah, Y shape, yeah, simply presentation as Y shape, yeah, can be found in our body in order to respond to a foreign object. Yeah, remember that actually foreign object here must be protein. Yeah, if introduce protein to our body, foreign protein, our body will create the antibody. Yeah, in infection is coming from bacteria or virus, for example. Yeah, they are carrying their own protein, right? They have specific. So our body recognizes foreign protein. So I will recognize, and then our body will generate antibody against it. Yeah, including what else? But must be protein based. Yeah, protein based. Sometimes you inject needle or jarum to your skin, but jarum is sterile. Our body will not create any antibody because there is no protein there. Yeah, so please just to remember that the foreign substance must be a protein. And then we have to understand the chemistry of antibody because we need to modify it in order to be acceptable for human. Yeah. So with regard to their structure, it has amino, yeah, carboxyl and so hydroxyl, sulfhydryl in their structures. Yeah. And then the monoclonal antibodies has what we call antigen binding fragment, we call it FAB, and also complement fixing fragment, yeah, FC. Yeah. And FAB fragment is responsible to specific antigen binding. Yeah. So this FAB is responsible to detect the foreign, the partner, the pasangan protein, yeah. Karena antibody is very specific, yeah, to certain antigen only, yeah. And FC fragment binds to the effector cells, yeah. That one actually to produce in vivo biological response. So again, we get to structure of monoclonal antibodies. Yeah, it has two antigen binding site, yeah, or epitopes, yeah, that can recognize antigen, yeah, and each subunit, yeah, antibody contains two light, yeah, chain, yeah, and two heavy chains, yeah, and then the variable domains, all antibodies contain amino acid sequences located both heavy and L chains. Yeah, so check later. We get to the structure, and then the heavy chains determine the class of antibody itself. Yeah, so the heavy chain will classify them into what we call gamma, yeah, IG, yeah, IgM, IgA, IgD, but this one in symbol, yeah. This one gamma, mu, alpha, delta, and epsilon. Yeah. So, but in the scientific purpose, they use IgG, IgM, IgA, and also IgD as well as IgE. Okay. And then you see the class, yeah, the class of monoclonal antibodies, yeah. They have IgG, yeah, the heavy chain, we call it gamma, yeah, IgM, IgA, IgD, and GE, yeah. So they have different molecular weight, yeah. IgA actually dimer, yeah, dimer of monoclonal antibodies. They have specific function actually, yeah. So I use IgG, IgM, IgE. Actually, they have specific. So later maybe you can just check what is the function of each, yeah. So this simplified presentation, yeah, of immunoglobulin, yeah, monoclonal antibodies, yeah. So they have heavy chain. The longest one is this one is actually heavy, yeah, heavy chain here, yeah, and I have light chain. Yeah, and this one is FAB. Yeah, they can recognize the antigen. This one is actually the function is for in vivo pharmacological action. Yeah. So because of structure of monoclonal antibody is quite big. Yeah, we call it macromolecules. Yeah, it is macromolecule because of its molecular weight is very huge. Yeah, compared to chemical drug. Yeah, so let's say IgG. Yeah, its molecular weight is 150 kilodalton. Yeah, for chemical drug, the molecular weight just only in Dalton something. Yeah, not. Kilodalton, not reaching kilodalton. You can play with the structure of monoclonal antibodies by reducing their size using two kind of enzyme, papain, and also pepsin. Papain actually from papaya. So papain can cut this hinge of monoclonal antibody to produce these two FAB, produce less molecular weight, and pepsin can cut at this position, and just follow your knowledge. Actually, you can actually cut the structure, even though the structure is quite big. You can cut them using two enzymes, papain also pepsin. So because the structure is very huge, consists of amino acid in their structure, and as we know, the monoclonal antibody is produced originally from mouse, so we have to understand each sequences of the amino acid structure. So actually we can replace the mouse part of the monoclonal antibody with a human part in order to be accepted by human body. So this, you see, this engineering on it. This we call it monoclonal antibody engineering. If you're interested, student, to the student interested to do more on this monoclonal antibody, actually there is engineering on it, to replace one amino acid with other in order to be accepted by human body. And then after the successful engineering, modification of the original monoclonal antibody from mouse, because originally it is produced from mouse. Later, we will have different kind of monoclonal antibodies. You see, the murine antibody is derived from mouse. This 100% from mouse. This one, this they label with O. And then they do some modification on this near to FAB fragment, with human antibodies. So replace this part becoming chimeric monoclonal antibody, about 60 to 70%. And then later they try to replace with 95% human sequences becoming zoo, humanized. And also to they replace all monoclonal antibody with human part will be human. Will be fully human. We call it dynamic with U. We can actually recognize that this type based on their name. You can check later. Whether they are coming from chimeric, humanized, or fully human. You can recognize them based on their name. So like this. This murine antibody, this originally from mouse. We call it the name is MOMAP, suffix. The what call the penghujung, nama dia tu ada MOMAP. And if they are chimeric, ZMAP. So at size ZMAP example. If it is humanized antibody, meaning about 90% of the structure coming from human amino acid, will be ZMAP. And if 100% human replaced by human antibody, will be MOMAP. So this actually they modified. This one can be used for therapy, yeah, but this one cannot be used for therapy. Next, how to produce, yeah, how to produce monoclonal antibodies. It can be produced using in vivo production using mouse acid test, and also can be used using in vitro, yeah, using mammalian cell culture, bacterial, transgenic animals, and also transgenic plants. Let's check on this interesting production using in vivo method. So in this process, if you want to produce monoclonal antibodies against certain cancer, yeah, we can just isolate the cancer cells from human. Yeah, we can introduce it, yeah, inject it into mouse. Yeah, what happened? Yeah, because the cancer cell is carrying protein, yeah, specific protein that is considered as antigen by mouse. Yeah, and then the immune system will recognize them as foreign substance, and of course there is response from mouse itself to generate antibody. Yeah, the antibody will be generated against that cancer is very specific. Yeah, and then where actually the antibody will produce in spleen, spleen cell of the mouse. Yeah, and then the spleen cell will be removed. Yeah. So meaning the spleen cell is able to generate antibody against that cancer. Yeah? And then after that, we from other side of the table example, we obtain myeloma cells. Yeah? So the spleen cell is considered as, if you are familiar with cell culture, the spleen cell can be cultured. Yeah? We can culture any cell from all part of our body, from your skin, from brain, bone, lung, spleen. What else? We can culture them outside. Yeah? We can just take, you can, if you're able to cut your, organ, you can culture actually. Yeah. But the nature of spleen cell is mortalized. Mortalized means just we can able to culture them for 2-3 subculture passage. Yeah? I mean, just within 1 week, the cell will be die. Yeah? And then, of course, we can stop our, but we don't want to be happen. Yeah? So, in order to extend the life of spleen cell, we obtain myeloma cell from mouse tumor, but we can actually purchase it from cell bank, no problem. And this myeloma cell is actually immortalized. Yeah, we call it immortalized. Can be cultured continuously. Yeah, without having death during the culture. And then the spleen. Yeah, the spleen cell and also the myeloma cell will be fused. Yeah, we culture them together in the blood. Yeah, and we call it hybridoma. Yeah? If you're familiar with hybridoma terms, actually hybrid. Yeah. Hybrid actually mix, including car, hybrid car, they are using battery and also petrol, hybrid. Sometimes the human can also be hybrid. No, just, I'm not sure, maybe the temperamental hybrid, for example, merged together in one people. Can be another study for that. And then, so now we have hybrid or fuse, and then during the merging, or merge or fuse of spleen cell hybridoma, some of cells are not meshed, they are not combining, So in order to kill myeloma cell from the culture, we can add drug actually to kill myeloma cell. Actually, we don't want myeloma cell to grow in our culture. You can add drug. So just only a successful fused cell will be cultured in future. Then each hybridoma is isolated. Later we can isolate that cell and allow them to grow. To produce monoclonal antibodies. And each monoclonal antibody is screened for its ability to attack original cells. So we can isolate the monoclonal antibody, yeah, try to culture them in other site. Yeah? So this one is really good to cell culture. Yeah, I hope you have some idea on actually how cell culture to be conducted. Yeah, and then later the desired hydroma cell are injected to mouse again. Yeah, actually to check to produce in large scale. So this is actually the production of the monoclonal antibodies using in vivo technique. Yeah. So like this, antigenic antigen can be cancer cell. For example, you want to produce antigen again lung cancer, for example. Yeah, we can just isolate the you can take it from human. Yeah. And inject into this mouse. So mouse will generate antibody in their spleen, right? Spleen cell. So we kill. Yeah, of course we need to sacrifice the animal. Take the spleen. Yeah, spleen we culture it. Yeah, we culture it on this plate. Yeah, and then this spleen cell, we know the nature of spleen cell is mortalized. Yeah, it can just only few can survive only for a few days. And then this we can obtain myeloma cell. Yeah, myeloma is immortalized. Yeah? It can survive for a long time. Yeah? And then we fuse them. Yeah? Fuse them in the same. Yeah? Actually, this two different cells we culture at the same plate. Yeah, they will fuse. Yeah, will merge together, and then will produce hybridoma cells. Yeah, and then later we can select. Yeah. Using drug to kill unsuccessful fuse cells, and later we can get pure or pure culture. Yeah, so this is really the simplified technique. Yeah, how to produce monoclonal antibodies. And then so we get to their name. Yeah, monoclonal antibodies you can obtain maybe in the market later. For example. Prefix can be varies. Yeah, prefix I think the awal nama can be apapun boleh. Yeah, and then target. What is the target of the that antibody? Yeah, like viral they use vir. Yeah, the source later on. Yeah, you can see from human U or mouse a rat something like that. And yeah, the suffix must be MAB. For example, at Sezimet, sample app can depend on the company, manufacturer, what kind of prefix they want to use. CI, this cardiovascular. This one for cardiovascular. XI refer to chimeric. Chimeric is about 60 to 70% of the monoclonal antibodies human part. MAB is a must. Yeah. So now I think we can recognize any product, or drug or whatever with suffix MAB, meaning they are monoclonal antibody. And then we see the milestone, with regard to development of antibody drug conjugation, for drug delivery. Since the introduction of magic bullet by Popplich 1913, there are some research has been done to conjugate, to check whether we have theory, right? We have theory. So everyone got theory now that specific cancer will produce antigen that can be recognized by antibody. So some trial has been done using methotrexate, the link it antibody, but this trial was failed. And then they tried to again what else? Some using drug. They're also using doxorubicin. But not successful. And in 2000, they tried to produce Myltap. This one is using doxorubicin. but it was withdrawn, yeah, from the market, from the not from the market, from the clinical trial, because failed, yeah, to work. What is the problem with Meltac? Yeah, Meltac because of premature release. Yeah, it is successful. Yeah, it was successful to combine with the drug, but when we introduce to human through parenteral administration, the drug is released during the travel. Yeah? So when they inject the drug, not being released at the site of the cancer, but it was released during the transport. Yeah, that one cannot be used. Yeah, because we don't want systemic effect. Yeah, and then yes, actually already reached the market. Yeah, but maybe not really widely used. Yeah, so 2010, my talk withdrawn, yeah, from the market. And then some research has been done and successful. Yeah? The first ADC drug was approved by FDA as a trace. Yeah? This one is for non-Hodgkin lymphoma, yeah, and also Kadcyla, yeah, approved for breast cancer to the 2013. Yeah. If you go to FDA website, you can check actually what is the current development with other type of ADC drug. Yeah? Go to Biologic, yeah, you can just see search ADC drug. Yeah, you can check actually. I mean, the research is not stopped here. Yeah, it's not stopping here. Yeah, currently many research, yeah, has been done by many countries, many companies actually to develop this ADC, but we are talking about the approved one. Yeah, the approved drug in the market is Atretris and Kadcyla so far. Yeah, some drug and still undergo clinical trial. Yeah. And this actually drug called other than Atretris and Kadcyla, yeah, this Besponsa, yeah. Besponsa was approved for 2017, yeah, by Pfizer. Yeah, this one just approved for certain area only. Yeah, call it for certain disease. We call it orphan medicine. Yeah, that can recognize C22 positive vessel. Yeah. So this one is also available, but I didn't mention it before. Yeah, but this one is just only for specific disease. Yeah, this one also using ADC approach. So maybe big question again, yeah, before we start with ADC. Yeah, what makes cell surface antigen suitable for antibody targeting in oncology? Yeah. Because an ideal expression profile, yeah, of abundant, yeah, the much expression, yeah, of the homogeneous antigen on external surface of all tumor cells, yeah, for multiple tumor types, yeah, with the majority of patients for each tumor type and absent from normal tissue. So this very, very important. Yeah, it must be specific only on certain area. Yeah, area here is on the cancer sites only. Yeah, but must be free. Yeah, absent from normal tissue. If not, the drug will reach normal tissue as well. Yeah? It can kill a normal tissue. Yeah, we don't want that to be happen. Yeah, so this is actually the basic theory. Yeah, regard to the development of ADC, yeah, in cancer therapy. Okay, again, let's check about the challenge, yeah, to treat solid tumor. You know, solid tumor. Yeah? Solid tumor is very difficult to treat because it has high interstitial fluid pressure. Yeah, I mean, even if you able just to apply directly, yeah, let's say the tumor is outside, you apply directly to the drug, yeah, directly on maybe like topical application, yeah, but the drug maybe cannot enter because of high interstitial fluid pressure. Yeah? Maybe the drug not able to reach, yeah, so all cell surface, yeah? And it has cell density, high cells. That's why difficult. Yeah, that's why for cancer therapy, chemotherapy with drug normally taking long time. Yeah? And also excessive deposition of extracellular matrix and also physical barrier compositomal proteins. Yeah, so that's why this approach is quite promising. Yeah, in order to reduce the burden or maybe suffering. Yeah, for the patient. We know that the cancer patient actually they are suffering. Yeah, they are suffering not only against cancer, they are suffering also against the drug that we prescribe to them. Yeah? So this approach is actually, to me, is kind of very useful, yeah, for the patient. And then the conjugation of antibodies, yeah. So drug targeting and delivery using antibodies has been most useful in this field, chemotherapy, because some what called drawbacks, yeah, or disadvantages of anti-cancer drug. Yeah, see, anti-cancer drug high toxicity, yeah, and they frequently have low therapeutic index. Between therapeutic index and toxic dose is very narrow. Yeah. High toxicity means must be very toxic, yeah, very toxic. If you given to human, the human itself can be killed. Yeah? So we don't have any option. That's why we still give it to the patient. Yeah? But what we discussed today is just close discussion. Yeah? So then to what called to discourage, patient to get the treatment using chemotherapy. Yeah? So this one is related to the role of IDC. Yeah? You see this effect, yeah? And also this dose. Yeah? So by using ADC in the treatment, we can actually expand, yeah, the therapeutic windows, yeah, between the toxic effect and also we get to the toxic and also we get to effect therapeutic dose. Yeah, so we can expand the therapeutic windows by using this approach. So now we move to the component of antibody drug conjugation. Yeah, so what we should have, we have to have this monoclonal antibody. Yeah, we have chosen already. Yeah, what kind of antibody that can recognize, yeah, specific antigen produced by cancer cell, yeah, for breast cancer we recognize HER2 protein, yeah, and for non-Hodgkin lymphoma CD30 or maybe CD22, yeah. So we have to have a specific monoclonal antibody, and then to link actually, yeah, how to conjugate drug. This monoclonal antibody is a drug. How to combine them? Yeah. Yeah. So this linker must be specific. Yeah. Meaning they will stable during the traveling of the drug to the target site. Yeah. So there's some linker that can be used. Yeah. And then after reaching the target site, reaching the cancers, the linker should be cleaved. Yeah. Should be cleaved, or some linker are not cleavable. Yeah. But will be digested by the biodegradable letter, yeah? All together, yeah? Of this component will be for non-cleavable linker. We'll check it later, yeah, to get more understanding with regard to this composition. So the concept of ADC, yeah, see this cell, yeah, these cells, yeah. Let's say this one is a breast cancer cell. So they have a HER2 positive, yeah. Normally for breast cancer patient, they will check antibody titer, yeah. So whether they have HER2 positive or not, yeah, in their blood, yeah. Then the cell surface of this cancer cell produce HER2 specific protein, yeah. And then certain antibody will recognize that antigen, yeah. They will reach this site and they will carry the drug, yeah. So by using this approach, it will increase drug delivery. Increase drug delivery meaning the drug only will be released at a certain site. To reach receptor, what can say that can to reach receptor. Similar to other type of drug, Actually, the drug what we take, and also if we compare with the amount of drug that can bind to receptor, actually the portion of the drug the drug that we need actually very less, compared to the drug that we take through oral, because let's say we take sample paracetamol. Paracetamol, one tablet is I think 650. Actually, how much of the paracetamol itself needed actually to relieve the pain sample? Maybe just only few micron. Whereas others, the others will be go throughout the body. Will be, I mean, of course, it will increase the work load of our liver in order to metabolize it, and also increase the workload of the kidney in order to dispose it. So by using this ADC, actually, it can increase the drug delivery. I mean, the amount can be, reach the target, and reduce normal tissue drug exposure, because we don't want them to be released during the traveling, So there is no effect to tissue, I mean other tissue or normal tissue, and then it can improve therapeutic windows. So now we get to the composition again. You would like to develop ADC, so it should have it has monoclonal antibodies, and should have linker, and also the drug itself, So each component should have a specific what you call properties, like monoclonal antibody should be selective, affinity and less monogenicity, and so on. They should have kind of properties. And linkage itself must be stable in circulation. So there is no enzyme, pH, or environment that can actually break them or cleave them during the travel. And should be stable in product storage, meaning when we store it, the drug before we give to the patient must be stable. They will still bind with, still do their job, to combine antibody and drug. Not maybe during the storage they are not working, they release it, right? So when we give to patient, the drug no longer to be in ADC form. And then trigger to release payload inside tumor cells, compatible for conjugation, of course. And then the drug itself must be high potency. High potency means must be very toxic. That's why for cancer, actually the drug that we give it is very toxic. Very high potency to kill cancer cells. Stable, must be stable. Low immunogenicity. And this actually other properties. So we can actually determine what kind of drug is suitable to be used. And when we get to immunoconjugate, we can also conjugate other substance. As we know, to treat cancer, can use whether we want to use drug, chemo, or radio, right? Radiotherapy, and radio actually very effective, to kill cancers. So radio isotope, radionuclide also can be combined with monoclonal antibody in order to be used in ADC. So can use a radio nuclide, also a protein or monotoxin, and also we can use drug. So not only drug, a toxin, radio isotope can also be conjugate to antibodies. So let's say for radio immunoconjugate, you can use beta emitting radioisotope, yttrium 90, or iodine 131. It is very effective. By using technology, you can actually combine this radio isotope with monoclonal antibodies. Yeah? So later we can use it to kill cancer cells. Yeah? This is a sample. Yeah? 131 iodine. Yeah? Using this one is yttrium. Yeah? Okay. And also radiolabeled antibody can be used to detect the location of the tumor, because the radio sort of is very sensitive. We can actually scan our body to localize tumor. Because the tumor is specific to produce antigen. We can bind antibody with radioisotope, inject to patient, and we'll go directly to the cancer location, and whole body can be screened, it can be scanned, scanning through the scanning machine. What is the name of the scanning machine? So we can localize where actually the cancer position, before we do operation of sample, throughout the body, whether it is in the lung, brain, whatever, we can check. So toxin also can be used, like toxin actually is very toxic, right? But if you're able to manage them, to be released to the certain, cancer location will be useful. So meaning need to do research on that. Need to be research on that, and many other toxin actually. We know that toxin is we have to avoid toxin, right? Any kind of toxin we should avoid. But maybe they are toxic again. Maybe they are very useful to treat certain cancer. We don't know. That's why we need to do research. Yes, in normal life, whatever toxin we have to avoid, right? But actually, we can utilize them to treat cancer. So the challenges is how to deliver them to the target site. And then this antibody drug again, so actually our topic is regard to antibody drug. So this one is very useful. Check. Then some structures of the drug. Why not all drugs can be conjugated? Actually, it is depend on the ability of linker, can bind to the drug at the same time they can bind to antibodies. So example, the drug that has been studied for ADC, doxorubicin. This one is not successful. Doxorubicin, and caliciamycin. This one is similar drug just now. Maybe I wrongly mentioned. Actually, for myelotag is not doxorubicin. My caliciamycin. Maybe in the previous slide I mentioned it was doxo. Not doxo. Caliciamycin. This caliciamycin is very toxic. Compound. And next one is auristatin, and also myostatin. But the success, this myelotag was not successful because of premature release during the development. But currently, some company still doing because of the effectiveness of, the activity of caliciamycin is very promising. Yes, not stopping, like myelotag has been withdrawn 2010. So, but they're still working on it to improve the delivery. And as a trace, using auristatin, Kadcyla. So what we have at the moment, Azathris and also Kadcyla. So this Azathris detect CD30, this is produced by non-Hodgkin lymphoma, and Kadcyla detect HER2 protein. So this comparison between Kadcyla and acetris. Yeah. See, this Kadcyla, approved 2013. Yeah, acetris 2010. Actually, my view on the screen is blocked by some this button. Yeah? And then so okay. So this different the differences between Kadcyla and acetris. Yeah. So monoclonal antibodies here they use humanized IgG. Yeah. This one is using chimeric IgG produced by CHO cells. Yeah. And target antigen. Yeah. This one is HER2. Yeah. This one is CD30. What else? Patient selection must be patient with positive CIS1. Yeah. This one positive for CD30. Yeah. And what else? The fresh. Yeah. This we get to the medicinal chemistry. Yeah. The comparison between the drug and antibody. Yeah. This 3.5. This one 4. Yeah. And the drug that being used, drug payload is, we call it maytansine or the M1. Yeah. So the mechanism of action of maytansine is microtubule inhibitor. Yeah. This part of the DNA disease. Yeah. So it can kill. Of course, they will affect the cell cancer to growth. Yeah. This is monomolecular auristatin. Yeah. It also working on microtubule disrupting agent. Yeah. Working on DNA. And linker. Yeah. Linker. They use this one is MCC linker. Yeah. This one is they use valine citrulline linker. I will check later. Yeah. What is MCC? What is valine citrulline? Yeah. And finished product. It is in lyophilized in dry form. Yeah. This one also in lyophilized because it is biologic product. Yeah. Biopharmaceutical product must be in dried form. Yeah. Cannot be in liquid. Yeah. Lyophilized is actually a product of freeze drying. Yeah. So we have to reconstitute. Yeah, we have to add water for injection before we inject to the patient. Yeah, and we get to the dose. This one. Yeah, this one the dose. Yeah, finished product half life about 36 months. This one. This is comparison. Yeah, between the two successful ADC that is available in the market. So this one the mechanism. Yeah, how they are working. Yeah. So this cancer cells, it's cancer cells, yeah. So they have antigen, surrounding, yeah, and then we introduce this basic understanding, yeah. But we are not talking about this very specifically, yeah. But what happened? Yeah? So surrounding this cancer cell, there is antigen. This is our antibody. Yeah. So how their work. Yeah. So then we can release here. Yeah. Can release the drug and enter the cell. And at the same time, the ADC can also internalize into the cells. Yeah. And then because of different condition between this endosome and also lysosome, the drug will be released, and then this free drug, now we have free drug here, free drug from this surface. Also drug will be internalized, maybe the drug will be released on the surface of the cancer cell, and drug will be internalized later, and bind to the target. The target is DNA, through microtubule binding, inhibitor. Of course, it can kill the cancer cell later. Okay, just go acid label linker, and so they are sulfate linkers. And another one is non-cleavable linkers. So acid label linkers because of different pH, in endosomal and also lysosomal. So because of acid, the drug will be released, and also the sulfate linkers because of this called enzyme, and peptide linkers will be released by this enzyme cathepsin, and non-cleavable linkers, the drug will be released after internalized of ADC, yeah? And it will undergo catabolic degradation. Okay. So this is the structure, yeah, of the linkers. Yeah? Can use this one cleavable linkers, yeah, non-cleavable, yeah. Non-cleavable linker can be a valence citrulline, yeah, hydrazone linker. This one acid sensitive, yeah. This one disulfide based on glutathione sensitive, yeah. This actually non-cleavable linker, yeah. Okay. So this is the complete composition, yeah, of the acetris, yeah. They have drug, payload, yeah, drug is the drug, yeah, and they have linker here, yeah. What else is the antibody that can only recognize CD30 antibody. This Kadcyla, yeah, again, this what call this using caliciamycin. This, I think this one is still called clinical trial. Example, yeah, can check in the FDA website, yeah, to check what is the status, yeah, of this ADC development. So another approach in ADC is using antibody directed enzyme product therapy. Yeah? So we can actually introduce prodrug. Yeah? Product, the drug itself is not active. Yeah? So when we introduce to the patient, the drug will be later convert. Yeah? Like this one using ADAP, yeah? Antibody directed enzyme product therapy. So we use a pyrimidine glucuronide. Yeah? So when we introduce to the patient, yeah, and because the enzyme, yeah, it will be converted to pyrimidine, yeah, and then the drug will be active against cancer cells. There's some process, yeah, involved in the administration, yeah, of the ADC. Yeah? So what happened? Yeah, we get to efficiency, yeah, of the administration. Yeah? If we introduce it into patient, yeah, how much ADC reach the tumor, yeah, need to be studied. Yeah? How much, yeah? And after that, how much ADC binding to the tumor cells, yeah? And then next, how much ADC internalized into the cell, and then how much of that will be cleaved need to be studied, and also how much drug reaching the target, yeah, and that's why. So this what called a step need to be monitored, yeah, in order to check, yeah, to ensure the efficacy of ADC, yeah. So some factor, yeah, the percentage, yeah. So what we expect is must be 100, yeah, 100%, but maybe in the what a real finding, maybe not. Yeah? So this advantages and also disadvantages. Yeah? For the conclusion, yeah. So the basic actually the basic fact actually, yeah, because of cancer cell produce antigen, yeah, specific antigen, then that antigen can be recognized, yeah, by specific antibodies, yeah, and then the specific antibodies can be conjugated, yeah, with drug, yeah, the very potent drug binding together using linker, yeah, and then we introduce it into patient, and the patient will what call the drug will go to directly, yeah, to the site of the cancer. And then because of different condition, whether it is because of enzyme, yeah, surrounding the cell, or maybe because of pH, the drug will be released, yeah, and later on the drug will be free, yeah. And if they are free, they will do their job, yeah. Whether they are, yeah, depend on their mechanism, yeah, where actually whether to do microtubule, yeah, or maybe other kind of activities can be also on cell membrane, yeah. It depend on the mechanism of action of each drug. I think that's all. So, thank you very much. I will give it to Mr. Moderator. Stop the share.
Thank you, Prof. Taher, for your explanation. For the next agenda, we are going to for next session is Q&A. Guest lecture is our participant are welcome if there is anything you want to ask by raising your hand, raise feature, or through the chat column. Please, does anyone to ask you about antibody, drug conjugation, and casatrophy? Participants are welcome if there is anything you want to ask question by raising your hand or through the chat column, please. Does anyone of you want to ask a question?
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Thank you, Mr. Dici. I want to ask to Prof. Taher about the monoclonal antibody. How about the stability issue for monoclonal antibody? As we know that protein is not stable in maybe heat or other thing that really critical for the protein. Thank you, Mr. Taher.
Okay, thank you very much, Taufik. Yeah. So yes, with regard to the stability of the monoclonal antibody, because monoclonal antibody is protein-based, they are not stable. So with the stability issue during the storage, that's why the product must be stored in lyophilized form. Yeah? I mean the right form. Yeah, right form. Of course, the storage normally they put in a refrigerator or low temperature, yeah? But not frozen. Yeah. So if they are in right form, they are more stable. Yeah, because of monoclonal antibody consists of amino acid that can oxidize and also can be oxidized by other agent. Yeah? So and also called if they are in water, maybe they are not stable as well. Yeah. So the storage must be in the right form and low temperature. Yeah. So when we want to use it, just add water for injection and then give it to patient. Of course, the preparation must be finished as soon as possible. Yeah. Otherwise, if the packaging is just only single dose. Okay. That's all. With regard to the stability to overcome the stability of similar to insulin as well. Insulin is protein based, but insulin nowadays available in liquid form.
Thank you for your answer, Prof. Anyone to ask question? Participants are welcome to ask your question if there is anything. Does anyone? Okay, Prof. I will ask you something about antibody drug conjugation. Maybe you can explain the advantage of the antibody drug conjugation in cancer if it was compared to the small molecule.
Yes, actually the approach of ADC, we don't want drug to be produce systemic effect. Yeah, meaning untargeted target. Yeah. So if the drug is in just in conventional formulation without ADC, so meaning it will affect healthy cell as well. Yeah, because the drug will be give it to the patient and produce a systemic effect. But by using ADC, maybe the dose will be reduced. Yeah, dose can be reduced actually. Yeah, as we compare with paracetamol just now. Yeah, paracetamol is dose is about 650. So actually, how much receptor really need? Yeah, maybe in micron, micro size. So that's the advantage. Meaning it can reduce the toxicity of the drug itself. Yeah, and also it can also because the toxicity. I mean. The dose maybe not the same. Yeah? Sample for if you're able to make ADC for doxorubicin, sample doxorubicin, how much per conventional administration, but using ADC the dose maybe very less. Yeah, very less compared to just in conventional formulation. Yes, that's all. Yes.
And so then the advantage of this antibody drug conjugation in cancer therapy, is it very important? Yes, sir.
Yes, yes.
Okay, thank you, sir. Okay, participant, if there anything you want to ask, last maybe. Does anyone you want to ask something about this guest lecture, especially on, with the topic antibody drug conjugation and cancer therapy, of course.
Or maybe this one is quite topic is quite advanced for them. What actually? Which year is it, Adam? Tahun berapa? Tahun?
Tahun.
Semua tahun. No, because maybe it is quite what called advanced for them. So I hope that actually it is not beyond your expectation.
The participants are first grade, second, and then last semester, sir.
Yeah. But at least they have some. I get some insight or idea with regard to the development of new, anti-cancer therapy. Because we have to face the bitter fact actually when we treat the cancer therapy, the cancer patient using a drug. Yeah? We have to we have to telan, maksudnya, even though it is pahit, yeah, but we have to try to save maybe our family, neighbor, or whatever, to survive against. But still consume or to use the drug, even though the side effect is very harmful or very bad to our body.
Minimize the toxicity.
Yes.
Does anyone you want to ask something about antibody in cancer or anything else about drug design maybe?
I think so because I'm a pharmacy lecturer. You can ask anything.
Anything.
Yeah, anything related to pharmacy. Don't ask me what's with regard to economy, how to increase what call market value of certain community. But it's okay, no problem. I think they what calls got something lah from my lecture. Yeah.
Okay, thank you, sir. Thank you, Professor Dr. Erwin Bahtiar. Dear colleague and friend, that is agenda that we have present to you today as we have finished our lecture. We would like to thanks again to Prof. Dr. M. Muhammad Tahir bin Bahtiar, PhD, for being a presenter or speaker in this guest lecture. Hopefully the knowledge he conveyed can be useful for all of us. Amin ya Allah ya Rabbal Alamin. So then, let's close this guest lecture with the word of Hamdalah and Kafaratul Majlis together. Alhamdulillahirrahmanirrahim. Subhanakallahumma wa bihamdika.
Okay, thank you very much. I would like to apologize for any mistake or whatever during my lecture. Maybe we can jumpa di lain waktu ya. Terima kasih.
Yep, amin.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Prof.
Welcome. Bye.
Prof. Tahir.
Salam.
Ya, silakan bagi peserta partisipan yang belum mengisi daftar hadir, nanti daftar hadir itu